Welcome to Science Views from the Valley. I'm your host, Dr. Kelly Kassan. Science Views from the Valley is a monthly program that will explore interesting science topics and how they relate to the San Luis Valley and the Upper Rio Grande region. I would like to thank everyone today for tuning in on this wonderful Christmas Eve. Today's topic is bioluminescence and biofluorescence. These two phenomena are how animals and other organisms can glow in the dark. The word bioluminescence comes from the Greek word bio, which means life, and the Latin word lumen, which means light. Bioluminescence resembles fluorescence and phosphorescence, but it is very different from these two phenomena. Bioluminescence is the creation of light as a result of a chemical reaction within the living organism. No light source is needed for this substance to glow. Phosphorescence and fluorescence involved a temporary absorption of light, so a light source is needed for the substance to produce the glow. Once the light source is removed, this substance will eventually lose its glow. The difference between phosphorescent materials and fluorescent materials is that phosphorescent materials often have a slower emission of the absorbed light than fluorescent materials. No phosphorescent organisms occurs in nature, though quite a few organisms will exhibit biofluorescence. All plants, for example, contain a fluorescent pigment called chlorophyll. Chlorophyll is a green pigment in plants that is responsible for absorbing light energy during photosynthesis, the process plants use to produce sugars. When chlorophyll is exposed to light, the pigment reaches an excited state and releases the energy to the plant's reaction center. This will start the chemical reactions needed to produce the chemical energy molecules that will be later used to produce glucose in the second stage of photosynthesis. But if chlorophyll is not contained within a plant or some other photosynthetic organism, then the pigment will release the light. This fluorescent light will be red instead of green. You can see chlorophyll fluorescence at home. Grind a few spinach leaves in nail polish remover. Nail polish remover is almost entirely acetone. Use enough nail polish remover to produce a dark green solution. Strain this green solution through a coffee filter or cheesecloth to remove the fiber and other plant solids, and then place this filtered solution in a tube or a small glass container. If you shine a flashlight through one side of the glass container and look at the solution at a 90-degree angle from the flashlight, you should see a red-colored solution. If you look at the solution from a different angle, the solution will appear dark green. This is an example of biofluorescence. Other examples of natural fluorescence include South American tree frogs and in sharks. Some South American tree frogs produce a skin secretion that will glow a bright green under UV light, otherwise known as black light. Frog researchers hypothesize that this fluorescence plays a role in camouflage or in communication between individual frogs. Cat sharks, otherwise known as dogfish, have biofluorescent pigments in their skin that emits a bright green glow when exposed to UV light. Different species of these small, bottom-feeding sharks have a different biofluorescent pattern. Male and female sharks of the same species also have different biofluorescent patterns. These pattern differences indicate that the biofluorescent pigments are used in shark communication and reproductive behavior. Scorpions are also known to be biofluorescent. Most scorpions have fluorescent chemicals in their exoskeleton, and under UV light, these chemicals will fluoresce a green or blue coloration. Scorpion hunters and researchers use headlamps that emit UV light in order to find scorpions. Here in southern Colorado, we have a very common scorpion called the striped bark scorpion. This scorpion will also fluoresce an almost radioactive green color when exposed to UV light. When the weather warms up, try looking for these scorpions under wood or rocks at night while using a black light to find them. The most well-known of the fluorescent molecules is the green fluorescent protein, or GFP, as it's more commonly known. GFP was first isolated by Japanese scientist Osamu Shimomura in 1962. He was studying the crystal jellyfish, a species that lives off the west coast of North America. The crystal jellyfish actually produces two light-emitting proteins, the fluorescent green GFP and a bioluminescent photoprotein called aquarin. Aquarin produces a blue light when it reacts with calcium ions. The gene that produces GFP has been used in genetic engineering, where it is extremely useful as a reporter gene. A reporter gene is a gene that reports that it and its accompanying genetic sequences have been successfully integrated into another organism's genetic material. When the GFP gene and the accompanying genetic sequences are successfully integrated, all of the cells with the gene will glow an easily recognizable bright green color when it is exposed to UV light. GFP's utility extends to medicine, where it is used to track cancer cells and viral infections. Because GFP has so many applications, Osamu Shimomura won the Nobel Prize in Chemistry in 2008, 46 years after his initial isolation of the protein. The GFP gene has also been inserted in living organisms for artistic purposes. One of the first of these organisms that were genetically engineered to glow green was a white rabbit called Alba, a project that was developed by Brazilian artist Eduardo Katz and French molecular biologist Louis-Marie Hudibina. Taiwanese scientists in 2006 developed green fluorescent pigs, which are called Noels, using the GFP gene. Since the discovery of GFP, other genes that produce a fluorescent light have been discovered. These include the red fluorescent protein, or RFP, which was isolated from a mushroom coral, and the fluorescent blue drompa, which was isolated from a stony coral. These genes, like GFP, have been used to produce fluorescent animals like glowfish. You may have seen glowfish in pet stores. These fish are usually zebrafish or tetras that have been genetically engineered with one of these fluorescent protein genes. Their color depends on which fluorescent protein gene was used. Now that we have talked about biofluorescence, which requires a light source, usually black light, in order to trigger the emission of light, what about animals that can glow without a light source? This phenomena is called bioluminescence and almost always involve two proteins, the enzyme luciferase and the molecule luciferin. Despite the sinister sound of the names, both names are derived from the Latin word lucifer, which means light bearer. Bioluminescence has been studied since ancient times. The Greek philosopher Aristotle observed that bioluminescence was a cold light, which means a light that does not produce heat, in 350 BC. Many of you are aware of fireflies, the small flying beetles that flash their abdomens on and off at night. That's one example of bioluminescence. But many different organisms have a way of glowing in the dark, including bacteria, protists, fungi, snails, insects, centipedes and millipedes, and even worms. So how do these animals produce light within their bodies? Luciferin, when it reacts with oxygen, produces the light. The reaction of luciferin and oxygen is accelerated by the enzyme luciferase. Enzymes work by binding to substrates, in this case luciferin, and placing the substrates in a position that allows the chemical reaction, the making and breaking of chemical bonds, to occur more easily and quickly. The color of the light that is emitted as a result of this chemical reaction depends on the structure of the luciferin molecule. Dozens of different types of luciferin molecules have been identified. The majority will produce a blue-green glow when it reacts with oxygen, but luciferin molecules that produce red, blue, and yellow light have also been identified and isolated. How this chemical reaction occurs in a living organism is still not completely understood. What we do know is that fireflies, arguably the best studied of the bioluminescent organisms, supply oxygen needed to complete the reaction through a small tube in the abdomen called the abdominal trachea. The abdominal trachea transports the oxygen to the light organ, which is called the lantern. Fireflies are able to control the amount of oxygen that flows into the lantern, allowing different species of fireflies to have different flash patterns. The purpose of bioluminescence is as varied as the number of bioluminescent species themselves. Several squid species use bioluminescence as a camouflage. They are able to control the brightness of their illumination in order to match the brightness of their background, which hides them from predators that are looking down upon them. Several species of shrimp and marine fish use bioluminescence as a way to startle their predators, giving them enough time to escape being eaten. Many cephalopods, these are squid and octopi, use bioluminescent molecules as a smokescreen to repel or distract potential predators, much like other squid and octopi use ink. Some squid will deliberately break off portions of its arms, which will continue to twitch and give off a bioluminescent light. This distracts potential predators as the squid swims away. The sea firefly, a small marine crustacean, will dart away from predators, leaving a cloud of blue bioluminescent light in order to confuse them. Another interesting way that bioluminescence is used as a defense is seen in phytoplankton. Some bioluminescent phytoplankton are spit back out by their predators, presumably because the presence of these phytoplankton will cause the stomach of the predators to glow. This makes them vulnerable to larger predators. A type of phytoplankton called dinoflagellates will shine when they detect predators, possibly to attract other larger predators that would eat the predators that would eat the dinoflagellates. Some predaceous deep-sea fish have evolved a countermeasure to this defense by having a black stomach lining that prevents the glow from the bioluminescent food from attracting larger predators. Another function of bioluminescence is warning coloration, a phenomenon known among biologists as aposematism. Some animals display distinct or bright colors that serve as a warning to potential predators that they are foul-tasting, toxic, or have dangerous appendages like spines or stingers. Poison dart frogs are an excellent example of this phenomena. They are very brightly colored with yellow, orange, blue, or red markings and secrete a potentially fatal toxin through their skin. Studies of millipedes, which are known to taste bad, found that predators will often avoid millipedes that exhibit bioluminescence, but will attack non-luminescent millipedes. Jellyfish are also believed to use bioluminescence as warning coloration, since all jellyfish are equipped with stinging cells that can maim or kill a predator. The most common purpose of bioluminescence is to attract mates or prey. Female fireflies use their ability to flash to attract potential mates at night. The pattern of the flash indicates which species the female is, so a male of the same species will have the same flash pattern, and they can find each other. But the fireflies are carnivores, and some females will mimic the flash of other firefly species in order to attract and eat the hapless males that come near. Another species that uses bioluminescence as a lure for prey is the deep-sea anglerfish and the dragonfish. These fish species do not make luciferin or the enzyme luciferase themselves, but they acquire bioluminescent bacteria which will reside in the facial appendages called esca. These symbiotic bacteria include Bioluminescence fischeri and Photobacterium. Approximately 10 million bacterial cells per milliliter are needed in order to produce a glowing lure in these fish. These symbiotic bacteria are hypothesized to be bioluminescent in order to disperse and find new symbiotic hosts. Though I have been focusing on animals, be aware that we also have bioluminescent fungi. Fungi species are hypothesized to be bioluminescent in order to attract grazing animals, which will eat the fungi and later excrete the spores. This allows the fungi to increase its range. Examples of bioluminescent fungi are found in Colorado. These include the common honey mushroom, which can be found near the base of old aspen tree stumps, and the aspen oyster mushroom, which is also found near dead aspen trees, but usually in mountainous regions. Though bioluminescence is seen in living things as simple as bacteria and as complex as animals, to date no truly bioluminescent plant has been found in nature. Throughout human history, bioluminescent species have been used by ancient civilizations. The Romans rubbed the bioluminescent slime of jellyfish on their walking sticks, which allowed the individuals to use them as walking sticks and as torches. Indigenous tribes in Indonesia used bioluminescent fungi as flashlights when they were walking through dark forests. Fireflies were collected in jars and used by miners as safety lights. Since bioluminescence produces a cold light, the use of this natural light prevented the ignition of underground gas. Bioluminescent species have been used in military operations as well. Angel's glow was described during the Civil War. Angel's glow is the result of bioluminescent soil bacteria known to have antimicrobial properties. During the Civil War, medics discovered that some soldiers that had these glowing wounds healed more quickly and cleanly than others. This was a result of these bioluminescent soil bacteria. A German U-boat in World War I was sunk in part because of bioluminescence. The U-boat had passed through a bloom of bioluminescent dinoflagellates, leaving a glowing wake that allowed the Allied forces to track and attack the submarine off the coast of Spain. This risk of having naval submarines and other marine devices accidentally revealed by bioluminescent bacteria was noted by other military. Research into bioluminescence as a way to detect enemies and to prevent the accidental bioluminescent reveals continues today. Sea fireflies, a small bioluminescent crustacean, were gathered and dried by members of the Japanese military during World War II. These dried crustaceans were used as a source of light so the Japanese soldiers could read maps during secret operations with a low risk of being detected by their enemies. In 1954, fighter pilot James Lovell, who would later become an astronaut and participate in the Apollo 13 mission, experienced a malfunction in his instrument panel while he was flying off the coast of Japan. He was unable to see in the darkness or use his instruments to find a safe place to land. He was saved by noticing a glowing green streak in the sea, the result of the U.S. carrier Shangri-La passing through bioluminescent plankton. Lovell used the trail in order to land safely on the carrier. Though no bioluminescent plant occurs in nature, scientists are currently genetically engineering plants so they will exhibit bioluminescence. They hope by doing this, they can produce a type of environmentally safe energy in the form of glowing trees that would replace street lamps. Bioluminescence can also be used to monitor water contamination. The bioluminescent bacteria, Bioluminescence fischeri, which I mentioned is used by anglerfish to create prey lures, is also used by humans to monitor water contamination. When water is polluted, these bacteria will decrease their light output. A device that measures the light output of these bacteria is already on the market for use in measuring toxicity in drinking and wastewater. In other science news, we have only one astronomical event that will occur in the San Luis Valley in January. This will be the Quadrantid meteor shower, which will reach its peak on January 2nd and 3rd. This meteor shower is thought to be the remaining debris of the now extinct comet 2003 EH1. An extinct comet is one that has expelled most of its volatile ice and has little left to form a tail. The remains of extinct comets, which is usually a large rock or a rubble pile, can resemble asteroids. The difference between an extinct comet and an asteroid is their orbits. Comets typically have eccentric orbits, and these orbits will remain eccentric even after they become extinct. Asteroids typically have more stable orbits. Scientists theorize that asteroids that have eccentric orbits are in fact old extinct comets. Approximately 6% of near-Earth asteroids are thought to be the remains of extinct comets. I hope you enjoyed learning about living things that glow today. My next show will be on January 28th, and I will discuss how different organisms stay alive and warm in the winter months, or in frigid habitats. As always, I welcome any science questions you may have, and will include them in future shows. Email your questions to spiderwomankrza@gmail.com. Please stay warm and have a very Merry Christmas as we count down to the end of 2020.